4 billion years ago, primitive Earth was a violent, inhospitable place. The surface was defined by intense heat, constant volcanic eruptions, lightning strikes, and unfiltered ultraviolet radiation. People have long tried to explain how a barren rock produced living organisms. The theory of special creation relied entirely on mythology, offering no testable mechanism. The Cosmozoic, or panspermia theory, suggested life arrived on meteorites from space, but that simply moves the problem to another planet without explaining the actual chemical origin. The cyanogen theory proposed that Earth's extreme heat fused carbon and nitrogen together. It was an early attempt to apply chemistry to the problem, but it failed to account for the actual complex building blocks we know make up living cells. It is also easy to confuse modern chemical evolution with the ancient theory of abiogenesis, often called spontaneous generation. That older theory claimed that fully formed living organisms, like frogs, snakes, or mice, could simply materialize out of non-living matter, like mud or rotting wheat. Louis Pasteur definitively proved that impossible. He boiled syrup to kill existing microbes, placing it in flasks with curved swan-like necks. The glass trapped airborne dust. Because dust couldn't reach the sterilized syrup, no microorganisms grew. Life only arises from pre-existing life. Since complex organisms do not spontaneously pop into existence, the very first cells had to be built from scratch. It required a slow, highly specific molecular assembly line operating over millions of years. That assembly line is known as chemical evolution, or chemogeny, formulated independently by biochemists Alexander Oparin and J.B.S. Haldane. The most critical condition for their hypothesis, and a frequent marker on your board exams, is that primitive Earth had a strictly reducing atmosphere. The air was rich in methane, ammonia, hydrogen gas, and water vapor. Crucially, there was zero free oxygen. Any free oxygen would have immediately oxidized and destroyed the fragile chemical bonds needed to build early molecules. Without oxygen tearing things apart, extreme thermal energy drove carbon and hydrogen to react. They formed saturated and unsaturated hydrocarbons. One molecule of CH reacts with another CH to yield HC triple bond CH, known as acetylene. The reactions continued, building slightly larger chains. Two CH2 groups fused to create ethylene containing a double carbon bond. As the Earth eventually cooled, steam condensed into rain, creating a hot primitive ocean. Water molecules reacted with those hydrocarbons to form oxyradicals. When you introduce ammonia into that mix, the nitrogen locks into the structure, synthesizing the very first simple amino acids. As macromolecules accumulated, they clumped together. This diagram shows a coacervate. For your exams, it is a colloidal droplet of complex organic molecules surrounded by a water shell. They absorb nutrients and divide by budding, but lack genetic material. To take the next step toward actual life, those coacervates needed instructions. When a coacervate successfully enclosed a self-replicating nucleic acid, like a primitive RNA strand, it became a protocell, or eobiont. Think back to the atmospheric conditions we established earlier. If you trapped methane, ammonia, hydrogen, and water vapor in a highly energetic environment for an entire week, what specific complex molecule would you expect to form? The answer is you would find amino acids. Enveloping those newly formed replicating molecules inside a lipid membrane marks the exact threshold where non-living chemistry organized itself into biological life. The Oparin-Haldane theory made chemical sense, but it required physical proof. In 1953, Stanley Miller and Miller-Urey built an apparatus to simulate the precise conditions of primitive Earth. This diagram illustrates their closed glass system. At the bottom, a flask of boiling water represents the hot primitive ocean, continuously cycling water vapor upward into the system. The vapor travels into a larger gas chamber containing the reducing atmosphere. For board exams, you must memorize this exact gas mixture and ratio: methane, ammonia, and hydrogen, locked in a 2:2:1 volume ratio. To provide the necessary activation energy, they inserted 2 tungsten electrodes that fired continuous 75,000-volt sparks, simulating prehistoric lightning storms. The gases were then passed through a condenser to cool, mimicking rainfall. Miller let this synthetic storm run for 7 straight days. When he analyzed the liquid collected in the U-trap at the bottom, he found sugars and simple amino acids, specifically glycine, alanine, and aspartic acid. The chemical evolution hypothesis was verified. Let's lock this pipeline in. History gave us theories of mythology, space spores, cyanogen heat, and spontaneous generation. Science gives us a verified chemical pathway. Simple inorganic gases fused into hydrocarbons. Those bonded into amino acids, which clustered into coacervates, and finally enveloped RNA to become a protocell. When you get tested on this, remember the 2 biggest constraints. The atmosphere was completely devoid of free oxygen, and the Miller-Urey experiment relied on a precise 2:2:1 ratio of methane, ammonia, and hydrogen. Mastering this sequence secures your board marks and reveals the exact chemical code for how a sterile, volcanic rock assembled the first living cell.